Welcome everybody. My name is Tony David. I'm the Vice Dean for Psychiatry. Welcome to the 18th Paul Janssen Lecture. The lecture is named after Paul Janssen, who was a brilliant pharmacologist from Belgium who discovered a number of very important psychiatric drugs and set up the company in his name. But this is a purely educational event, and the only specification is that the speaker should be an international leader of the field, tick, that they should be talking about something with at least a vague relevance to schizophrenia, tick, and they've got to be a brilliant communicator, tick. So we're in for a great treat. So the lecturer is Chris Pantelis, and he is the scientific director of the Melbourne Neuropsychiatry Centre, and he holds the, get this right, professorial fellow position at the Florey Institute for Neuroscience and Mental Health. And he also heads the Adult Health Rehabilitation Unit at Sunshine Hospital. So I visited there several years ago now, and it's just that we don't have hospitals here called Sunshine. And it really was a beautiful place, brilliantly architecturally designed, and you felt better just going near there. So Chris actually is from Australia, of course. He graduated from Melbourne, but then he came to London. He worked at the Royal Free Hospital and then at Charing Cross Hospital, and got interested, I guess, in schizophrenia research. And we got to know him at that time, and we were both very interested in neuropsychology, which was a really expanding field. So he went back to Australia and really took the field by storm. One of the things about Australia is that we like to sort of patronize our cousins down under, but they really are showing us the way, not only in the cricket, but certainly in terms of early intervention in psychiatric disorder. It's really from Melbourne that that concept has taken such a powerful hold and has led the way. And so what Chris has done is combined that therapeutic zeal with real research excellence and expertise, and a lot of it's to do with neuroimaging and seeing what happens to people in their brains as they go through these transitions. So he's a very highly cited author, has over 600 publications. One of his most cited publications is a 2003 paper in The Lancet, which was really set the tone for the kind of longitudinal studies, neuroimaging studies, tracing people from as they go into psychosis or avoid psychosis. And I guess that's the theme for today's talk. So it was a brilliant paper. It was decades ahead of its time. I know a few people in this audience were co-authors. It was great for this institute to be aligned with such a great piece of work. So without further ado, I'll hand you over to our speaker, Chris Pantelis.
Thank you very much, Tony, and for those very kind words. It's fabulous to be back in London. I'd spent 10 years here, and I've got a number of close friends and colleagues, and it's fabulous to catch up with them. Most recently, we lost Ian Everall, who sits here and has come and is now the Dean here, and it's been fabulous working with him, so it's nice to see him here. I'm going to talk about trajectories because I think that the way forward in terms of thinking about the disorder is to think about it longitudinally. And I think I want to add to that the notion that risk and resilience might be important, looking at things like risk and protective factors. So most of our funding comes from our NH and MRC, and indeed we've been continuously funded since 1994, soon after I got back from Australia. In the talk today, I'm going to focus on trajectories and then talk a little bit about, and really what is an emerging field in terms of thinking about resilience factors and thinking about perhaps the neurobiology of resilience, which I think has not really been pursued although a number of people are now interested to take this forward. Acute relapse, I think, is another area that we really need to focus on, and the picture that I showed you at the beginning was really to remind us that psychosis is a very dramatic event, and when young people develop what is in effect like a psychotic storm, it's very traumatizing, stressful, both for the individual and for their families, and a lot of work really needs to go into helping those young people come to understand and move forward in a very positive way, and that's a hard thing to do. In thinking about acute relapse, one of the areas that we've got very interested in has been neuroinflammation, and I'm going to touch on some of the work that we've been doing there, looking particularly at microglia and more recently the complement system. And given the nature of what this lecture is about, I thought it was important to think about treatment. And one of the directions that we've been taking recently is looking at how can we modify trajectories. And that's leading to some interesting translational work to animal studies and other things that I'll talk about. I'm a clinician. I work clinically. I look after patients with the most severe forms of schizophrenia. And throughout my career, many of the research questions I've been involved with have really been generated by observations at the bedside. And I'd like to think about not only bench to bedside research, but the importance of bedside to bench. In other words, the value in those clinical observations leading to important questions that can be translated to the bench, and then we can go back again. Obviously, I'll be talking about imaging. We've done a lot of neuroimaging, but increasingly we've moved to the right side of that top row there, looking at genetics, looking at neuropathology. Indeed, the work with Ian Everall was really focused on neuropathology. We're working with the Monash Institute of Pharmaceuticals to identify new treatment options, and we're developing new animal models to take forward. When we think about psychosis, it's important to place it in context. And that context is that it occurs in young people. It occurs in adolescence and early adulthood. And when you think about it, that context is very different to what we see at the other end of the age spectrum in older age, where you see predominantly neurodegenerative disorders. And therefore we need to be thinking, well, what's the neurobiology of normal maturational processes? Because likely they play a part in how the illness is evolving. They play a part in perhaps how the illness is interfering with that maturational trajectory. Indeed, what's interesting, when you look even at neurodegenerative disorders such as frontotemporal dementia, and this is a study undertaken by Dennis Velakoulis in our centre, he looked at all the published cases of FTD. We then looked at it according to age of onset of symptoms. What was striking was that those with the earlier age of onset, particularly in the 20s, were significantly more likely to manifest psychotic symptoms. In other words, there's something about the youngish brain, young adult brain or adolescent brain that makes it more susceptible to manifest such psychotic symptoms. So that kind of context is important because the maturational changes that we see, as many of you will be very familiar with, includes a gradual thinning of the brain cortex, and you see this here in this paper, another highly cited paper by Nitin Gogte, as the brain there on the left is going from bright colours to blue. There's a thinning that starts at the back of the brain, moves forward and downward, and finishes in association cortices, including dorsal prefrontal, superior temporal, gyral areas, key areas implicated in schizophrenia. So that context means that there's a very dynamic system, a changing system during adolescence and early adulthood. In parallel with that, there's myelination occurring. Whilst most of the myelination occurs in the first few years of life, importantly, it continues into adulthood. Indeed, some of the more recent data suggests into the 30s, and that's particularly in association cortices such as the frontal lobes. So these processes are very dynamic. They're occurring at an important stage of development and at a time when psychosis itself is evolving. And these are the data from Huttenlocher showing the notion of synaptic pruning occurring in adolescence as children move into adolescence and adulthood. And this is particularly in regions such as the frontal cortex, those regions that are changing dynamically. In effect, then, we have a very dynamic system. And when we look at data from schizophrenia, it's important to consider those findings, that data in that context. And if you don't do that, I think you don't get the right picture emerging about what's going on in this disorder. So I think it's important to go back in history a little bit and look at some of the early studies that might suggest there is evidence for progression as well. I think it's also striking when you go back to Kraepelin and you see that based on clinical observation, what Kraepelin was able to discern was that key regions, in frontal and temporal lobe areas, are likely to be impacted in this condition. That's quite extraordinary when you think about it, because they certainly didn't have the tools that we have today to explore that. And it hasn't changed very much. These are the key regions we still consider are important. And that was based on clinical observation, and stresses the importance of that. These studies were highlighted to me by Linda Lisi, some early longitudinal imaging studies using pneumoencephalography. I don't know if many of you, I know some of you will be familiar with pneumoencephalography, but basically you insert a needle into the spine, you extract a little bit of CSF, and then inject a little bit of air into the CSF space. You tilt the patient so that the air goes into the ventricles, place them on an X-ray table, take an X-ray, and then you use a ruler to measure the size of the ventricles. Very painful procedure, leads to headache, which can go on for days or weeks. And these people had it on 2 occasions, showing evidence for ventricular enlargement, which is certainly interesting. Of course, there were a number of interventions going on in those patients at that time as well. These 2 papers were key papers published in 1987 independently, but coming pretty much to very similar conclusions. So Robin Murray from here, together with Sean Lewis, discussed the importance, and based on the epidemiological data, and based on evidence from that early insult during fetal development increased the risk for the disorder, came to the conclusion that some kind of early insult was important, and that somehow during development, when systems that had been impacted by that insult appeared to be dysfunctional as you matured. And Danny Weinberger, looking more at neurobiological indices, came to a very similar conclusion. These 2 papers really drove the field for the next 2 decades. And the notion that I think was interesting here was that the notion that there was a fixed lesion. I would argue that, yes, I think they were really on the right path here, but the notion that the lesion necessarily occurred at a particular stage of development was probably not quite right. Indeed, insult at any stage of development, from first trimester right through to adolescence and beyond, can increase your risk for developing schizophrenia. The other important aspect to these 2 papers was that it spoke to the notion that environment was important, that there was an interaction between whatever was happening in the individual and the environment. And environment by environmental and neurobiological factors are likely to be important, including genetics. And I'll come back to that a little bit later. So the MRI changes. Basically, they demonstrated, or they have demonstrated, that key regions in frontal, temporal, and subcortical areas are involved in schizophrenia. And when we first started looking at our patients with chronic schizophrenia, we didn't use imaging, we used neurocognition, as Tony David had indicated. And here were some of the first studies we did in chronic schizophrenia. These were 30 years ago, collecting the data using the CANTAB, which had just been worked up, had just been established at that time. So we were fortunate to link up with Trevor Robbins and Barbara Zahakian, and to use those techniques in these patients. I went around and assessed as many of the patients at Horton Hospital as possible, and that led to a number of key findings, again demonstrating that frontal and temporal lobe regions were impacted in this disorder. And these are the key regions implicated. Dorsal prefrontal cortex involved in higher level mental functions, including working memory, strategic ability, etc. That's been a key and consistent region in this disorder. But other regions are equally important. Anterior cingulate, involved in attentional processes. The orbitofrontal cortex, when I started in this field, there was not much attention paid to this region, but very important in terms of social cognition, higher order social cognition, as well as tasks such as smell identification ability. And we undertook a series of studies using tasks such as smell identification to probe the integrity of this and other regions. So the frontal lobes are key, and not only are they the last to mature, they're also the last to evolve. And it's likely that this is somehow important, the evolutionary history of the development of the brain and what it might mean in the context of the emergence of schizophrenia. And this is a meta-analysis of voxel-based morphometry studies, picking up and highlighting frontal, temporal, subcortical regions. The regions we were a little bit surprised about was the insula bilaterally, which showed volumetric reduction. A key area where sensory information comes in. And interestingly enough, this is one of the regions that changes during transition and onset of the illness, and was, at least in our data, predictive of onset of disorder. When you do a similar voxel-based meta-analysis of voxel-based morphometry studies at earlier stages of the illness, and these are data from the Institute of Psychiatry, you see some similar regions involved, but certainly not as dramatic as what you see in chronic schizophrenia patients. And even these data point to the possibility of progressive change over time. White matter imaging has come to the fore in the last few years. About 10 years ago, we employed Andrew Zaleski. He was an electrical engineer, had no biological training. He'd been working on wiring, and we gave him the task of looking at the wiring in the brain. It was a bit of a gamble, I have to say, but he really did a fabulous job. He developed a tractographic method to look at the brain, statistical techniques to interrogate these networks, and this was the first paper published, showing, again, that wiring, white matter integrity was problematic in frontal, temporal regions, but also, interestingly enough, in more posterior regions in the occipital area. And this, I think, is interesting because it comes up again in early psychosis as well. More recently, we looked at white matter integrity in a much larger sample of patients with chronic schizophrenia collected across Australia. And whilst the picture on the right suggests that there are some key areas that are impacted in terms of tracks connecting different regions, I think the most compelling picture is the one on the left, showing decreased fibre integrity. This is a measure called fractional anisotropy, and it looks at the integrity of white matter, the directionality of flow of water in the brain. And what you see there, in effect, is that all brain regions are affected. White matter appears to be globally affected, or at least in this larger sample. And the recent publication by the Enigma Consortium is consistent with these findings. We've also looked at the possible genetic contribution to white matter tracks. And this was a study that appeared just a few days ago, looking at a gene that was that's been relevant to schizophrenia. It's involved in neuronal migration, synaptic pruning, deemed to be a plasticity gene. And we found, and it has both a protective and a risk haplotype. And the protective haplotype showed a significant interaction in terms of important white matter tracks with differences in both schizophrenia and controls. The importance of these data is that I think we need to look at it longitudinally, we need to look at it at earlier stages, and if it is a plasticity gene, then it may be that there are important interactions with the environment. So gene-environment interactions would be interesting to pursue here. So the data here are pointing to progressive changes, but certainly in chronic schizophrenia you see a number of brain regions impacted, and particularly not only in grey matter but also white matter. The possibility of progressive changes is what I want to show you, and how our data evolved, how we looked at how we came to the Lancet paper. I was fortunate to work with Dan Weinberger back in the early 90s, and that's where I started the imaging work. At that time, they just published this paper in the New England Journal of Medicine in twins discordant for schizophrenia. What they identified were two key features that differentiated these brains: the increased size of the ventricles that you see there, but also smaller hippocampi. And almost at the individual level, you could discern which twin had schizophrenia based on those indices. So it was a logical step for us to look first at hippocampal volume, important area of the brain involved in memory, behaviour, and linked to emotion. And when I got back to Melbourne, as Tony indicated, I started working together with Pat McGorry and Alison Jung. They were at the very beginning of their ultra-high risk for psychosis studies. They were just defining this syndrome. And this is a summary of the at-risk for psychosis syndrome, deemed according to attenuated psychotic symptoms or transient psychotic symptoms lasting perhaps less than a week, or state or trait risk factors such as schizotypal personality or a family history of the disorder. What was relevant was that almost 40% of patients of these individuals at high risk made the transition to psychosis over a 12 to 18 month period. That meant that we could scan them at the very beginning before they developed psychosis and follow them after, until they developed a psychotic illness, or waited for a 12-month period if they didn't develop psychosis and rescanned. So we were able to look at the pre-psychotic state, and we were able to work out whether we could identify a marker for the disorder. Indeed, our hypothesis was that hippocampus would be a marker that would pick up those individuals who would subsequently develop the disorder. We looked across stages of the illness, and this study involved almost 500 cases. Dennis Velakoulis traced all the hippocampi in this blind to diagnosis. He did it on 2 occasions so that we could be sure of the data, because it was contrary to what we had expected. What we found was that in chronic schizophrenia, as we predicted, there was bilateral hippocampal involvement. However, when we went back to earlier stages of the illness, first episode schizophrenia, the left hippocampus was down but not the right, and then in the schizophreniform, the coloured bars just next to that, there was no significant difference from control. So these bars are deviation from normal. And more importantly, when we looked at the high-risk individuals who subsequently developed psychosis, they did not show any hippocampal reduction. So surprising, and the data here suggested to us that we should look longitudinally. And that led to the paper in the Lancet showing progressive changes in grey matter a progressive loss of volume of grey matter in frontal and temporal lobe regions, including medial temporal lobe regions, as well as the cingulate bilaterally. Concurrently, we also measured cognition, and we looked at memory relevant to the hippocampus. Here in a paired associate learning task, which is sensitive to the integrity of the hippocampus, we showed that at baseline, individuals in their first episode of illness, and also we have data in our high risk, indicated that they were not impaired on this task. So not only was the hippocampus didn't appear to be involved, at least volumetrically, but also functionally it didn't seem impaired. At 7 to 10 year follow-up, what we identified, and published this recently, was that there was a significant deterioration in this ability, suggestive of progressive decline. And we're now looking at the hippocampus in more detail. There's a study that one of our PhD students, Cassie Wannen, has been undertaking, and showing that in schizophrenia there's a correlation between the size of the hippocampus and performance on that associative learning task. What's interesting about this task, so it's the ability to make associations with regard to information that's not necessarily related. What's interesting is that when you look at it developmentally, from a developmental perspective, this task matures very early. So kids perform very well on this task. So it's likely that by the time individuals who will develop psychosis come to that point, they're already matured in terms of their ability to perform the task. By contrast, when we looked at the neocortical changes, first of all, we see in this study on the right here that patients with first episode schizophrenia scanned over 2 to 4 years after onset of the illness. So they were scanned within the first few weeks and months, and then followed over 2 to 4 year period. What we see there in the middle row is that patients with schizophrenia are manifesting a dramatic volume reduction, or at least retraction of the surface of the neocortex in the frontal region, which is going twice the rate of what we see in normal controls. Indeed, when we map the rate of change in the patients versus the controls, it's twofold, not only in this region, but across the whole brain. We think that, or we consider that it's significant in the frontal regions because this is the region that's changing most dramatically at this maturational stage of development. So in effect, it's likely that there's an interaction between the age of onset of the illness and the nature of the abnormality that you might see, because potentially the onset of the illness interrupts that maturational trajectory. And indeed, we worked with Nitin Gogte to compare his childhood schizophrenia patients with those with adolescent and early adulthood, and I'll come to the childhood schizophrenia in a little bit. We also looked at function relevant to this frontal lobe region. We looked at spatial working memory. The 3 lines in the circle there are patients with chronic schizophrenia, first episode schizophrenia, and the high risk who develop psychosis. And on this spatial working memory task, they were showing a similar deficit. And over a 7-year mean period, we saw no further deterioration. So a fairly stable deficit present from before the onset of the illness. What's interesting is when you look maturationally at this task, by contrast to the previous one, what you see is that it's still maturing in adolescence and early adulthood. You can see here also that males do better than females, and that's probably because it's a spatial task. I'm sure if it was a verbal task, it would be the females. So it's nice to be able to do well on some tasks. So it got us thinking about trajectories, and we suggested that there were a number of potential trajectories that might be involved in this disorder. I've shown you evidence for neuroprogression in a loss of grey matter volume as well as deterioration in function. I've shown you evidence for potential developmental arrest. So I think the data on working memory suggests that the onset of the illness interrupts the maturational trajectory of that ability. But the line that I want to focus on is the one just below the normals, and that's neurodevelopmental lag. So the line at the top is the normal controls. The other important point with regard to this graph is that if you were to scan or to assess individuals at the time point where the three lines intersect, you might consider that these individuals have the same abnormality. Clearly, however, when you track them over time, they're on very different trajectories. And if you're looking for points of differentiation in terms of how people are trekking, you need to look longitudinally. So that's exactly what we did here. So these are data in childhood schizophrenia. We worked with Nitin Gogte and Judy Rappaport to look at structural covariance as a measure of brain connectivity. What you see in the graph on the right there are three lines. The red line are kids with childhood schizophrenia. They show an abnormality in this connectivity measure at age 12. They have a very severe phenotype as well. They look very similar to some of my treatment-resistant patients, and most of them are on clozapine, so very severely affected by the disorder. The line at the top in blue are typically developing children who show connectivity which is fairly consistent over the ages from 12 to 24. What I think is interesting is the line in the middle in black. That line is the unaffected siblings of those children with schizophrenia. And what that demonstrates is that an abnormality in the structural connectivity resolves by age 16. So by age 16, the unaffected siblings of the children with schizophrenia have normalized in terms of this this connectivity measure. You can see this also dramatically here in these movies provided by Nitin Gogte. The numbers represent the timing in terms of scans, dates of the years at which they were scanned, and the coloured areas represent grey matter loss. And the panel on the left, the childhood schizophrenia, the panel on the right are the unaffected siblings. By age 16, they've normalised in terms of the thickness across the brain. If you're looking for an endophenotype of schizophrenia, what's clear from these data is that if you scan individuals or if you scan the relatives at age 18 or 16 and above, you're likely to miss a potential endophenotype, which is only apparent at age 12 to 16. So that's an important consideration as well. What these data have taught us is that there's a potential that things which look abnormal can resolve, and it raises important questions about what protective mechanisms might be involved in that. It's also got us thinking about focusing in our high-risk population, not only at those individuals who develop psychosis, but also at those who don't. In other words, those who appear very similar to the pre-psychotic group, but who never develop a psychotic illness. And this is a study of white matter fractional anisotropy, looking at the integrity of white matter over a 12-month period and looking at the relationship to symptoms. So improvement in white matter integrity was associated with improvement in symptoms. So whilst much of the attention to date has been on those who develop a psychosis, actually I think we need to be focusing a little bit more on those who remain well and try and understand some of the mechanisms that might be involved to keep people well or to prevent the onset of the illness. So we can see here that the brain is changing in terms of structure in early psychosis, and that it's associated with functional changes. There's neuroprogressive changes in temporal, frontal, and as well as subcortical regions, and that we see evidence for an acceleration of normal maturational trajectories, which may have relevance in thinking about what drives that kind of change. The notion that there are improved trajectories means that we should start turning our attention to potential resilience factors. And looking at risk factors and resilience factors means we should start looking at things like genes, stress, drugs, and possible role of neuroinflammation or inflammatory processes. So, in thinking about trajectories, one of the things that we started to consider was what happens with acute relapse. So we've been looking at changes over a period of time, but what's clear is that with every relapse episode, what we see is a loss of function. People, after they have an illness episode and have a remission, they don't get back to their pre-morbid level of functioning. Presumably this means that there's some kind of neurobiological factor that might be involved in leading to this deterioration. But there's very little data to inform us in this regard. We looked at our data in terms of the longitudinal imaging. And here are the two panels show the superior temporal gyrus and the insular cortex. And we look at different stages of the disorder. T1 and T2 are the time 1, time 2 scans. We're looking at chronic patients. Those at high risk who don't develop psychosis is the second column. And then the two in yellow are the ultra high risk who develop psychosis, and then the first episode, psychosis, followed by chronic schizophrenia. Most of the action, it seems, at least in terms of grey matter, seems to be occurring within the first 5 years, 4 to 5 years of illness onset. And particularly at transition. And transition is a very dramatic event, as I mentioned earlier on. And it's likely that the brain changes are most dramatic when the illness itself is most dramatic, potentially. And when thinking about trajectories, whilst many of the longitudinal studies to date have used 2 time points, it's important to consider that that may not be enough, particularly when you have relapse and remission episodes. So, for instance, if you look at the cubic function there, and if you were to scan at the origin of that red line and then at the second point, you're likely to see a difference in whatever measure that you might be interested in. If, however, for the same individual, if you scan them at the origin of the blue line and then again at the second time point, you're not going to see a difference. In other words, the trajectory may be more complex. And indeed, we've demonstrated that there are differences in the way that things are changing in the brain in terms of both grey matter and white matter according to age. Surprisingly, in this study that Vanessa Cropley undertook, we saw that white matter changes were most dramatic later in the illness, and that the grey matter seemed to be changing most earlier on in the illness. So this timing of changes is something that we need to take account of as well, and that is likely to be impacted by the way that the brain is maturing. We looked in the literature to see what we know about relapse and remission in terms of neurobiology. And there was a paper by Garver published in 2000 that we thought was really interesting. And this paper showed that with psychotic relapse, he only looked at two measures, brain size and ventricular size. With psychotic remission, what you saw was increase in brain volume and reduction in ventricular size. It seemed that the brain was swelling, in effect. And with remission in the same individuals, he saw the opposite pattern. The brain size went down and the ventricles enlarged. So this very dynamic process over relapse and remission also suggested a very dynamic process in terms of the neurobiology, and meant that we really need to be thinking about how we're going to examine the brain longitudinally. We also had some data that was supportive of these findings. Here we looked at T2 relaxometry in the hippocampus. T2 relaxometry allows us to index a water signal, and we can look at it not only in the CSF, where you get a very bright signal, as you can see there, but we also looked at T2 signal in the tissue. We looked at the hippocampus, and in the high-risk individuals who subsequently developed psychosis, you saw an increased T2 signal. Are these data in support of what GABA was showing? We also looked at the ventricles. And this study was the same individuals in which we looked at the hippocampi. And we looked on the left side of that graph at just the individuals with schizophrenia or schizophreniform illness. And you can see there in terms of ventricles, it's larger in chronic, not as large in first episode. The schizophreniform people with less than 6 months of illness didn't show increased ventricles. What was really interesting, though, was the high risk, who subsequently developed schizophrenia. Their ventricles were reduced compared to normal. So is it possible that we're seeing this very dynamic change in ventricles and brain volume? And all of you will be familiar with this study published in Nature, a GWAS, the largest GWAS study published to date in schizophrenia. Huge number of patients and subjects involved in the study, and it picked up this very high signal reflecting immune function. So genes in the HLA area seemed to be implicated with a p-value of 10 to the minus 30. It's important to say, though, that the effect size was actually very small. There was a lot of excitement because dopamine and glutamate were also picked up in this study. But the immune signal, I think, is interesting. And if we think about the dynamic process that might be occurring with relapse and remission, one possibility is that there's some kind of inflammatory process occurring in the brain in people as they transition or as they have a relapse episode. So we looked at some inflammatory genes in high-risk individuals and looked at transition to psychosis, and we found that this cytokine was predicting those, so SNPs in this gene were predicting who developed psychosis. And we've subsequently been looking at the relationship to things like brain volume, etc., with an impact on ventricles. Given that background, we were interested to examine inflammation and inflammatory processes, because it's likely to be important in terms of the trajectories of possible change in the brain in this disorder. Indeed, we thought it might explain the grey matter changes that we were seeing. So we examined microglial activation. So the microglia are an interesting cells in the brain. They're derived from the periphery during the second trimester. They're related to macrophages, monocytes, and white cells. What you see there is a study in an animal model in vivo, using an in vivo technique. You induce a lesion just below the red line. And then you see in this time-lapse photography technique an infiltration of activated microglia. So the microglia normally are quiescent in the brain. They're surveying the environment. They have all their tentacles, out doing that sort of surveillance work. And then if they detect some kind of insult, they withdraw those, they become sort of amoeboid-like and aggressive, and descend to contain the damage, if you like. You can imagine that in patients with psychosis, where an insult may not be quite as clear, if these guys became active, they may do a fair amount of damage because they can strip away axons and synapses and dendrites. You can index this using a ligand with PET to identify the protein on the surface of these cells when they become activated. We used one of the original ligands, and there's been some criticism about this, and I know there's been a lot of really interesting work here from Oliver Howe's group in this regard, showing increase in these inflammatory cells in high-risk individuals. But the literature, when you review all of the studies, is sort of up and down. It's a bit unclear at the moment about what the microglial story might be. The consensus that's emerging, though, is that perhaps activation of these cells is not as we might have expected. So we undertook perhaps one of the largest studies to date. Over 70 individuals scanned across stages of the disorder. Marie Diabias was our PhD student, did really a fabulous job here. The upshot of our findings was that microglial activation was not evident. We didn't see evidence for microglial activation across stages of the disorder. Indeed, the individuals who were unmedicated, if anything, showed a reduced signal. And my question is whether, indeed, these microglia are behaving normally. If there's some kind of genetic abnormality, it may be that they're not behaving normally. Following on from the Nature paper, this paper came out in, I think, in 2016, which tried to interrogate that HLA region, the MHC region, which is so significant in the GWAS, and identified the complement system as important. And the complement seems to be relevant to the activity of microglia. And for this reason, we also examined it in our patients. What we did see in these same individuals was increased levels of C3, complement C3 and complement C4. And it was particularly at the earlier stages of the disorder. So is it likely that these might be involved in affecting some of the brain changes that we're seeing? Still, obviously, a lot of work to do, and we're just looking at the relationship to our imaging findings in terms of that. In the last few minutes, because I know we should be finishing fairly soon, I want to talk a little bit about how genetic analysis may inform novel targets, and then finish off with something on treatments. We started to get interested in resilience when we did this study in autism. So I'm not an autism researcher, but working with Stan Skafidis, an engineer who applied some of his analytical techniques to GWAS data in autism, we used a pathway analysis approach to look at whether we could classify individuals with autism versus controls. Indeed, we were able to, with about 72% accuracy, which is not that high, but it's not too bad, based on SNP scores, which included SNPs that were risk SNPs and SNPs that appeared to be protective in some way. So there was a balance between risk and protective SNPs. These guys in the middle, so you can see autism and controls were separated. These guys in the middle were the parents, and the ones in the middle there were siblings. And it's interesting that some of the siblings were scoring fairly high, and it raises questions about why they weren't diagnosed with autism. Just as an aside, it's important with these studies, because there's a lot of these studies going on currently, that when you're looking at prediction, it's important to take it back to the population. What's the prevalence of the disorder in the population? Because when you look at that and look at our data in the context of the prevalence of autism in the population the positive predictive value falls to just under 3%. So that's an important caveat that I think we need to take account of when looking at any prediction studies. Here were some of the more common SNPs that we'd identified. And I've mapped these on the right side in terms of risk SNPs and on the left side in terms of protective SNPs. And one of the areas that we became interested in, which showed both protective and risk contributions, was the metabotropic glutamate receptor 5, which modulates the activity of microglia. So we undertook a study looking at the brains of individuals with autism to look at the expression of GRM5 in the brain in the dorsal prefrontal cortex. There was reduced expression in those with autism. And in a parallel study using an mGluR5 knockout mouse, Liliana, our PhD student here, was able to demonstrate that reduced mGluR5 activity led to increase in microglia in the brains of mice. The link here is that this is potentially a modulator of the activity of microglia. We believe that in autism there's likely to be a subtype where inflammation is relevant. Looking at these kinds of data might point the way to novel treatments for the disorder. We undertook also pathway analysis in schizophrenia. And this picked up across 3 ethnic groups. We picked up 5 pathways that were common across those ethnic groups. The other important finding in this paper was that many pathways were affected, and it may well be that many pathways in the brain lead to schizophrenia as such. But we concurrently were looking at the ubiquitin system in the brain, and this is certainly something that Ian here will be able to tell you a lot more about than I, but we looked at the various components of the ubiquitin pathway. It's an important pathway in terms of removal of debris, if you like, from cells. And we see increase in ubiquitinated protein levels in the brain. And I show you this as an example of where we can take the data from genetics to inform us about pathways that might be affected, that can lead potentially to understanding some of the neurobiological changes that we're seeing. And we see that both in the data from autism as well as in the data from schizophrenia. Finally, modifying developmental trajectories. And are there not only treatments, I want to focus on some environmental factors. Sarah Whittle in our group has been involved in the adolescent development study, a large study looking at adolescents across development from age 10 to adulthood, and with serial scanning on that group. And in this study, what she did was to look at positive parenting behaviour, assessed using a problem-solving task, in which we looked at positive parenting and its influence on brain trajectories over the adolescent period. And we looked first at the impact of socioeconomic disadvantage. People living in areas that were disadvantaged showed anomalous brain development involving particularly the temporal lobe of the brain. When we looked at positive parenting, positive maternal behaviours, what we see is that that was able to ameliorate some of these trajectories over that developmental period. The importance of this, I guess, is that a change such as that's environmental in effect, a change in the way that parenting behaviour can influence the impact on brain development over adolescence and has clear implications in terms of treatment in disadvantaged areas. And finally, in terms of treatments, we've been interested in cognition as a key factor, a key deficit in schizophrenia. And it's been very difficult to treat cognitive deficits. We've tried various antipsychotic drugs in terms of amelioration of such deficits with very small effect sizes. And here we've linked up with the Monash Institute of Pharmaceutical Sciences. Indeed, we've just got a program grant to look at translation from the bedside to the bench, taking what we've found in human studies to animal studies to inform us about how we might go back to the human. And in this study, we looked at an MK801 glutamatergic model of cognitive deficit in the mouse. So you give MK801, leads to cognitive deficits, particularly memory, and then we sought to try and rescue that deficit. We looked at a number of antipsychotic drugs alone, including typicals and atypicals with no effect. And we added in a positive allosteric modulator of the M1 receptor, the muscarinic receptor, the muscarinic system obviously being important to cognition. It was the combination of that positive allosteric modulator with the atypical antipsychotics that led to a rescuing of that cognitive deficit. So potential implications in terms of what we might be able to do with combination treatments in our patients, particularly at a time when we're all talking about monotherapy. In fact, what we might need might be to judiciously use treatments that might target other kinds of symptoms. We're now working with the animal CANTAB. So I started off my career using the human CANTAB. There's now been an animal CANTAB developed, and you can see that here. The mouse approaches the screens and touches them with their nose to get a response, and then gets a treat at the end. So we're able to do paired associate learning, we're able to do reversal learning, and we're going to be using this in developmental models as a way forward to try and identify new treatments. So, schizophrenia needs to be considered within a maturational context is really the message I want to convey. That unless we do that, we don't get a clear picture of what's going on in this disorder. We really need to map what's going on in schizophrenia over time, not only from the onset, not only from just before the onset. We actually need now to do the studies, which are hard studies to do, right from birth through and follow. And there have been some really fabulous studies in this regard, but this, I think, needs to be the way forward. And if we're going to be thinking about treatments, if we're going to be thinking about modifying trajectories, we probably need to intervene that early. If we're thinking of remediating cognition, remediating some of the negative symptoms, then perhaps we need to intervene at a very early stage. Looking at gene-environment interactions is really a nice way to go. The importance of the adolescent development study demonstrating that an environmental change can lead to a change in the way that the brain is developing. For me, that was a very exciting study, and it means that we can develop strategies to help parents in disadvantaged areas to do better and to help their kids grow differently. Developing novel animal models. We're locked into a lot of the old animal models, the PPIs, etc., which are all good, but I think we need to start thinking in terms of longitudinal studies and animal work which takes account of developmental trajectories. Not an easy task, I might add. And in order to really get at understanding not only the neurobiology, but ways forward in terms of treatment, we need to bring together disciplines that are disparate. And it's a very exciting way to go. So when I started the work with autism, I was sitting with Stan Skafidis every Friday for about 5 years. We worked together. And that was fabulous because what it meant was that we could translate between the disciplines, which was necessary to do. I'm going to finish there. There's a lot of people to thank. This has been a huge collaboration over many years with a lot of people in the room as well. It's been fabulous to work with such great people. It's still an evolving story. There's still much that we need to do and much that we need to take forward. We need to change the way that this illness progresses over time because the outcomes for many of our patients are so dire. Not all, of course, but for a lot of them. And finally, I want to attract you all to Melbourne. It's a great place to be. Now, Ian left, so don't take him as an example, but it really is a great place. It's sort of the university's down there somewhere, but it's a great place where neuroscience has come together in one precinct. We've got the Melbourne Brain Centre, we've got the Immunology Institute, we've got the Florey Institute, we have our centre, we have neuropsychiatry. It's all come together. We have engineering, computational scientists, etc. It's a fabulous place to be, a very vibrant place to be in terms of where we are currently. And the important thing about it all is it's the people. It's not the place, in effect. I mean, the place is great to bring people together, but it is the people. And that's our group at Christmas last year. You'll see Ian in some of those pictures. We had a couple of program grants with Pat McGorry, and more recently, program grant with this guy over here, Arthur Christopoulos, at the Pharmaceutical Sciences group. Very exciting group to be working with. And, of course, we need technologies, because the new technologies that have been emerging have been really instrumental in helping us to take forward the field in ways that we might not have imagined 20 years ago. Certainly I didn't imagine 20 years ago. It's a very exciting time to be in neuroscience. Really exciting. There's a lot of fabulous things to do, and I hope some of the students might consider a career in psychiatric neuroscience, because it's just wonderful. Thank you.
Chris, thank you for a fantastic talk. Melbourne is a fantastic city. It's great the amount of neuroscience going on there, and the weather is much better than here. I've known Chris for about 20 years, and I had the pleasure of working with him closely for 8 when I lived and worked in Melbourne. And so I've heard him talk many times. And during that time, I've really enjoyed the maturation of this concept of trajectories, because I think it's really important. But every time I hear him, I always get really impressed by his grasp of the concepts and of the field. You know, for someone to be able to take data from clinical, cognition, imaging, neuropathology, neuroscience, genetics, and now therapeutic molecule identification is really impressive. Plus the wider integration of interdisciplinary research. In fact, Chris and I worked so closely with engineers that they made us adjunct professors of electronic and electrical engineering, which really made me smile, because I can just about change a plug. And so, you know, I think that Chris has actually crystallised at least 20, 25 years of wonderful research and just his impact on the field. So I'd like to thank Chris, and I'd like to ask you to thank me in thanking Chris and treating us tonight to a wonderful 2018 Janssen Lecture. Thank you, Chris.
Okay, thanks very much, Ian Everall, for that vote of thanks. Thank you for coming. There's a reception in the Education Hub, the Robin Murray area, to which you're all invited. Thank you very much.